First, I'll illustrate bonding and specificity in DNA. I made the phosphate backbone and started with deoxyribose. Each had one pipe cleaner, 5 beads, and 2 gold rings. I put 5 beads on each deoxyribose to represent our carbons, and a gold ringlet by the 3 and 5 prime carbons to help me remember where the phosphates are bonded. 1 prime carbon is where we will attach the bases. 2 prime carbon is where we can tell if it is DNA or RNA by whether or not an oxygen is present. In our case, there's no oxygen because we're making DNA. 4 prime carbon is where the 5 prime carbon branches off. Then I add a nitrogenous basis to the 1 prime carbon of deoxyribose. I'm using old Easter eggs as bases, with pink representing the complementary bases of adenine and thymine, which are bonded by 2 hydrogen bonds, and blue representing complementary bases cytosine and guanine, which are bonded by 3 hydrogen bonds. Since pyrimidines have 1 ring and purines have 2, we'll have the longer sides of the eggs represent the purines and the shorter sides represent pyrimidines.
Now if we look at a double-stranded DNA, each phosphate molecule is bonded to the 3 and 5 prime carbons, so the connections of the sugar-phosphate backbones are called phosphodiester bonds. Notice how one DNA strand has the oxygen pointing up in the deoxyribose and the other oxygens are pointing down. This resembles an anti-parallel structure. I chose Easter eggs in order to demonstrate that the purpose of hydrogen bonds are for specificity. However, this model does not illustrate how the structure of DNA is supported. Colored clothespins resembling our bases, the hydrogen bonds between the clothespins are not the force that holds the 2 strands of DNA together. Instead, it's due to the hydrophobic interactions of the uncharged bases. Since they are uncharged and hydrophobic, and the backbone is negatively charged and hydrophilic, the flat bases can lay horizontally and stack themselves in a space-efficient and low-energy manner. This is called base stacking, which creates more hydrophobic interactions in this inner area, making it much more stable. The negative charge of the backbone is destabilizing and hydrophilic, but the salt neutralizes the negative charge, making it more hydrophobic like the bases. This increases the melting temperature because more energy is needed to separate DNA that is made more stable with salt. To illustrate this, I'll push a pipe cleaner that is hydrophilic and saturated with water through the middle of an oil blot. The differences of polarity will break the oil blot. But if I take a pipe cleaner and saturate it in oil to decrease the differences of polarity, it's slightly more difficult to do. The major groove is a larger gap between the strands, while the minor groove is smaller. DNA is right-handed if it looks like a spiral staircase with the hand railing on the right side while walking upstairs, and it is left-handed if the handrail looks like it's on the left side while walking upstairs.